Okay, cool. Yeah, hello everybody. Good afternoon. My name is Florian Klimm from the Novo Nordisk Research Center in Oxford. So we are a partnership between the University of Oxford and the Danish pharmaceutical company Novo Nordisk. This is joint work with Maxwell Ruby and Robert Kitchen. And today I'm going to tell you how we can use CRISPR knockout screens to identify gene regulatory pathways. I should highlight we are all employees, so there's a potential conflict of interest, but I will be honest to you anyways. Okay, so what is CRISPR? Some of you might be aware of it. It's a genome engineering technique, which is quite influential and actually won the Nobel Prize in Chemistry in 2020. How does it work? So it takes a protein called Cas9, which can induce double-strand breaks in the DNA and so deactivate certain locations on the DNA. And then you can use the so-called guide RNA in combination with this protein to guide this Cas9 protein to specific locations on the genome and so knock out specific genes. This really has created a new era in functional genomics because it allows this knockout of specific genes at unprecedented scale, affordability, and reliability. And so we in Oxford use these screens to get a deeper understanding of the disease biology. And so in the end, it helps us to find better treatments for diseases. These CRISPR knockout screens are used in a wide range of different screening modalities. What we're going to focus on today are so-called survival screens. I will go very briefly over them. So essentially what you do, you take some cell line that you're interested in, you induce some lentiviral transduction to produce this single guide RNA in each of the different cells. I'm sorry. And then you have these knockout in different cells and you study how well these cells are able to survive. It gives us a measure of something called cell viability. And today I'm going to talk about some public available data set called DepMap, also Project Achilles. So what researchers did there is they did these pooled CRISPR knockout screens in approximately 1,000 different cell lines, most of them cancer, but not all of them. And then the readout is this cell viability that I just talked about. So it can be used to identify which genes are essential for the cell survival. So you can imagine if you knock out a really important gene like actin, the cell won't be able to survive, whereas some more peripheral gene, the knockout might actually not have really a strong effect in the cell viability. But I'm going to show you today that we can also use this data to interrogate gene pathways. So due to compatibility issues, you don't see my data, but you see at least my trend line. So you have to show some imagination now. So this is here on the horizontal axis, the knockout effect of one particular gene called PKM. On the vertical axis is a different gene called ENO1. And then when you're going to see this data, which you don't see, there are 1,000 data points, each of them different cell line, and it gives us the information how strong the knockout effect in each of those two genes is. And so again, each data point would be a different cell line. And then if we compute a trend line as a correlation, we find here that these two genes are highly correlated. However, if we take a different pair of genes, so now PKM and MPAC1, then we see they're actually not correlated, as you see in this orange line. So this gives us already some indication that some of these genes are highly correlated, whereas other pairs of genes are not strongly correlated or not at all. We can test this now for all genes. They did this knockout. So they did about 17,000 of these knockout screens. So they have 17,000 different genes in 1,000 cell lines. And so then we can compute this correlation matrix. And what we already observe is some kind of block diagonal structure if we reorder appropriately through hierarchical clustering. And when we now see this block hierarchical structure, that there are some blocks along the diagonal which are groups of genes which are very strongly correlated. And so they are encapsulating pathway information if we perform a gene ontology enrichment analysis. To go one step back, I mean, you might now say, well, that's just a correlation network. But what I would argue that we're actually going one step further to causality in these network analysis, because here we now really know which genes are knocked out, and it's not just a correlation. Or it's a correlation, but a correlation of a causal knockout. We can also zoom in more specific in subparts of these matrix. For example, one biological pathway we are quite interested in, glycolysis. And then what we can do, we can observe this matrix. And again, we do a block diagonal structure where we see there's mitochondrial glycolysis, which is strongly correlated with each other, and cytosolic glycolysis. And there we even have two kind of like sub-pathways, which are the early part and the late part. And if we now look at the KEC pathway diagram, this matches particularly well. One thing which we were surprised by, but it actually shows some influence, is the so-called proximity bias. So it just means this is again the correlation matrix. And now I order the genes not as earlier by a hierarchical clustering, but by their position on the genome. And what we observe is that they actually also here now along the diagonal, you have stronger correlation. And so this is likely the case if you knock out one gene, you have also a bit of a likelihood of knocking out the gene next door. So we might have to correct for this proximity bias. Won't go in detail here. We did a small correction of proximity bias, and as you're going to see in 2 minutes, it didn't help much. But what we do, we essentially just match the different distributions, and it didn't change our results much. So I'm not sure whether we should do better in correction or whether just this is actually bias is not really perturbing our results much. So now we want to check whether the network we create actually represents different pathways well. And so how can we do this? We compute something called conductance, which you might know if you are familiar with graph theory. So we can take in a network a subset of nodes, which are highlighted here in red, and we're gonna compute how well this subset of nodes is kind of like in the whole network represented. And so we do this by computing first the volume. So that's the sum of all weights of edges in the pathway. And then the cut size is the sum of outgoing edges in the pathway, which are highlighted here in green. And then we can compute the inverse conductance as this volume over the cut size. And so we can compute this now for all pathways we have in our data. So here I took all pathways which were in the KEC pathway annotation, and we find that there are many of them which are along the diagonal. So those are those pathways where the cut size is the same as the volume. But now, however, then there are also some of the diagonal, and those are the pathways which are actually strongly enriched in our correlation network. And then we can look at this for all the different pathways we have, these inverse conductance. And then you would see here now distribution with most of them having very low conductance. So they are not well represented in our network. However, some of them are really well presented in our network because they have a high inverse conductance. And that's one of these results which was first actually not nice, and we were kind of surprised by this. But I think what it just shows is that some of our pathways are really influential in the cell viability readout, whereas other pathways are not well represented in the correlation network that we computed. So then you might ask, well, correlation matrices, you usually would threshold to construct a network. How can we choose this threshold? And that's that we explored here. So on the horizontal axis, you have different thresholds for constructing our network. And then on the vertical axis, you have how good the enrichment of our pathways is as the mean inverse conductance. And first of all, what you see, there's kind of like a bimodal distribution. So it seems fine at about 0.5 and also a bit higher at 0.65. And then now the dashed line is this corrected matrix where we corrected for proximity bias. And you see, essentially, it doesn't change our result much. So again, I'm not sure whether it says our correction is not very good or actually we don't have to correct. Happy to discuss this later. So far, we have only looked at existing pathways. But of course, now we want to go a step further and maybe actually use this information to refine existing pathways. And so what we use here is a well-established technique, which probably a lot of you are familiar with, was also mentioned in the last talk, network propagation. Specifically, we compute the personalized page rank. What it is, it takes a set of seed nodes and it finds other nodes in our network which are close by. The nice thing about this personalized page rank is that it converges very quickly numerically, so it's quite easy to compute. First of all, to validate our approach, we want to do a cross-validation. So we took one CAG pathway glycolysis, and we always take out, so we do a leave one out cross-validation. So we take out one of these genes from the 67, we do the personalized page rank on the remaining 66, and we look what's the rank of the one gene that we removed. And we see that the rank is usually very low, much lower than the baseline, which you would see from 0.5. So on average, you would expect it in the middle of your list. Then something which I didn't talk about, there's an alpha parameter, which is a free parameter in this personalized page rank, and it seems to have negligible influence. And very interestingly, the best prediction is at the threshold 0.5 from our correlation network, which is very similar to the result we saw earlier, which is of course nice that these very distinct approaches align here in their outcome. To give you one example where we might actually use on biology, which we don't know yet, very well. We can take 3 genes, for example, lambda2, lambda4, lambda5 as seed nodes, apply our personalized page rank algorithm to find other genes which are close by in the correlation network, and we correctly identify other members of the regulatory RAG complex, so lambda3 and RAGA, but we also find other genes which you wouldn't have expected to be in any way involved in this pathway, for example, VPS41. And so that's, I would say, the point where it gets interesting because it's now a hypothesis generating tool. So I know it's an overused metaphor, but like if you think of ChatGPT, it gives you a lot of like wrong results, but it gives you also some like novel ideas. And I see this in a bit similar. So it gives you some ideas. You probably shouldn't trust everything that it predicts, but it's something to get you thinking. So to conclude, I hope I convinced you that we can construct these correlation networks from CRISPR knockouts, and they can be used to identify biological pathways. And then we can use network propagation, specifically personalized page rank, to get other genes of similar biological function to a set of seed nodes. Lastly, I want to say, if you're interested in Novo Nordisk, the work we are doing, I have a poster today, OA400, but I have a lot of very talented colleagues of other posters on the work we are doing. We do a lot of different things, trajectory of diagnoses, complex diseases, insulin-resistant knowledge graph. So all my colleagues would be very happy to talk with you about this. And also my colleague Joe is at the career fair on Thursday if you want to talk about opportunities. Yeah, we have job openings coming up all around the world, UK, India, China, and so on. Thank you very much for your attention.
We have time for questions.
Thank you. Thanks for a great presentation. I'm curious, how did you deal with off-targets?
Excuse me, Biff?
How did you deal with CRISPR off-targets? You already showed an off-target effect of nearby genes.
Yeah.
So how do you know what you see is not due to off-target effects?
Well, I mean, I would say since we get a pathway enrichment, I hope that's reality. But of course, there are probably more biases we can correct for. Oh, so I mean, those are the published screens by DepMap. So they did the experimental corrections they did themselves.
Other questions?